Hello and welcome. This video forms part of an instructional series covering the general use of Pion's MacroFlux, MacroFlux, and BioFlux apparatus with the Rainbow R6 concentration monitor and its counterpart software, AU Pro 7.0. In this video, I'll provide an introduction to the different items of hardware which comprise the MacroFlux and BioFlux apparatus. Here we have a diagram of one MacroFlux assay vessel when fully assembled. It is composed of two chambers separated by a lipid-coated PVDF membrane. The donor chamber, which is a 1000 ml maximum operating volume glass vessel as used for USP2 style dissolution apparatus, and a smaller, disposable plastic acceptor chamber, which is suspended inside the donor vessel from a custom vessel cover and fitted with an independently operated stirrer. Both compartments accommodate rainbow fiber optic probes for in situ quantitation. Typically, any sample being assayed for flux is introduced directly to the donor medium to commence the experiment. In the acceptor chamber, separated from the donor chamber by the lipophilic membrane at the base of the plastic insert, the appearance of the test sample is measured and values of flux are calculated from the observed rate of appearance. Pion's acceptor sink buffer is used inside the acceptor vessel to prevent the system from reaching equilibrium during the assay. Up to 4 MacroFlux assays may be performed simultaneously using an 8-channel rainbow instrument. A variety of different vessel covers are available to suit various brands of USP2 dissolution system. BioFlux uses the same setup as the MacroFlux, except in place of the USP2 vessel, a lower volume 500 ml vessel replaces it. BioFlux is typically performed with donor chamber volumes of between 200 and 250 ml. It uses a slightly extended version of the MacroFlux plastic acceptor chamber in order for the base of the insert to reach the donor medium. This version of the apparatus yields a more realistic dose-to-volume ratio than standard USP2 apparatus, and also conserves media. Laid out on the bench here, we see all the individual components of the MacroFlux and BioFlux apparatus. These are the 500 ml and 1 litre glass vessels which comprise the donor chamber for BioFlux and MacroFlux respectively. This is the associated plastic vessel cover. This particular type is intended for use with Wecker USP2 dissolution apparatus. Vessel covers for other types of dissolution apparatus are available. On top of the cover, we see openings for a rainbow fiber optic probe, a smaller cover for adding samples through, an opening in the center for accommodating the stirrer shaft, and lastly, a larger opening through which the acceptor vessel is inserted. Acceptor vessels for MacroFlux and BioFlux are disposable items and ship in packs of 8. The longer vessel is used for BioFlux applications, and the shorter for MacroFlux. Each plastic vessel has a PVDF membrane affixed to the bottom with a total surface area of 3.69 square centimeters. Before installation to the apparatus, the acceptor chamber membranes are painted with 50 microliters of GIT0 lipid solution and are filled with up to 16 milliliters of acceptor sink buffer. This is the flux stirrer head. On top of the device, there is a connector with a protective rubber cap to shield against liquids. It is important that this connector always has the cap fitted when a cable is not connected to the stirrer head. Getting liquid inside the connector will corrode the pins and render it useless. The stirrer head is connected by one of these grey cables to this stirrer control box, which is used to set the stir speed of all connected stirrer heads, usually 250 RPM. There is a small hole through the top of the stirrer head for accommodating an angled fiber optic probe. There is also a rack, which accommodates 200ml flasks in which to store the stirrers when they are not in use. Other important accessories include these 13mm diameter plastic spheres, which are used to set the stirrer height of the Airwecker bath appropriately for the BioFlux glass vessels. We also have J-style fiber optic probes, with their associated probe holder. These have an advantage over straight dip probes, as the horizontal orientation of the light path prevents the accumulation of undissolved solids and also the entrapment of air. Lastly, we have a smaller diameter, fixed 10mm path length fiber optic dip probe, which is placed in the acceptor vessel through the hole in the stirrer head. The height of the dip probe in the vessel may be controlled by affixing this metal ring to the probe at an appropriate height. In this video series, we will be demonstrating the use of the BioFlux kit with the Airwecker DT126 apparatus. But I should note that other types of dissolution testing apparatus are supported. Please contact your Pion representative for more information about using BioFlux or MacroFlux with other types of dissolution testing apparatus. This concludes our video overview of the BioFlux and MacroFlux hardware and accessories. The next video in this series will introduce the different consumable reagents which are required for performing flux assays.